And it's my pleasure now to introduce Dr. S.J. Gao, who's the program leader of the Cancer Virology program.
Good morning. It is my honor to present the Cancer Virology program. I have been in the program leadership position since July 2018. My expertise is in KSHV and AIDS-related malignancy. I have some leadership experiences in my previous institutions, and my main role is to provide scientific leadership for the program, facilitate inter-program interaction, and promote program growth. Some history of the program. Following the discovery of KSHV, a working group on AIDS-related malignancy was formed, which eventually led to the recruitment of Dr. Chang and Mo to the Cancer Center and the formation of the Cancer Virology program. Dr. Mo was named the inaugural program leader. The group later went on to discover a second cancer-causing virus, Merkel cell polyoma viruses. I was very excited to join and lead such an exceptional program. The program has 27 members, bringing more than 8 million fundings. 3 million is from NCI. Members of the program publish 470 papers. Many of them are from inter-program collaborations. In the last renewal cycle, the program was rated outstanding. The reviewer mainly recommended us to strengthen the interaction with the clinical investigators. We have increased our interaction with the clinical investigator, which has led to several new clinical trials in viral cancers. We have also taken advantage of our leadership role in the contribution of data set to TCGA and used them to identify new cancer-causing virus, as well as assess the presence of no virus sequences. We have made some adjustment to the program in order to be better cancer focused, as well as to be better aligned with the new vision of the Cancer Center. These include addition of 8 new members, 6 of them are new recruits. Showing here are 3 examples of the outstanding young investigators who work on humanized mouse model, EBV long non-coding RNA, and MCV oncology. The program has 4 integrated themes, which are to delineate the mechanism of viral oncogenesis, identify new therapeutic target and agent, develop new viral vector for cancer therapy, identify new biomarkers, and discover new cancer-causing viruses. I will present several vignettes from the program. The supported grant and the use of shared resources are shown at the bottom of these slides. In an effort to identify new circular cancer-causing viruses, in this intra-program collaboration, Dr. Chang and Mo's lab has identified new circular RNA encoded by EBV and KSHV. These new circular RNA are present in tumor sample. They are highly abundant and stable, implicating their likely important functions and possibility of using them as a biomarker. The group is working with the max cohort investigator to test this feasibility. This is a new publication from Dr. Shuda's group. In this collaborative study, Dr. Shuda's lab have demonstrated differentiation of Merkel cell polyoma cell, Merkel cell carcinoma cell into neuronal cell. The result of this study implicate that neuronal precursor cell could be the cell origin of Merkel cell carcinoma, and SOX2 is required for MCV-induced cellular transformations. In this new intra-program collaboration, Dr. Shear's group has developed a new air-liquid interface 3D culture for examining EBV replication and spread in epithelial cells. This new system is particularly useful for studying EBV-induced nasopharyngeal carcinoma and gastric cancers. Dr. Sarkar's group is interested in immunity and viral cancers. His group has previously shown that interferon-stimulated gene, OASL, enhance REI-mediated RNA sensing. In this new inter-intro program collaboration, his group has now shown that OASL inhibits cGAS-mediated DNA sensing. Since innate immunity is closely related to cancer inflammation and cancer progression, the group is now collaborating with other investigators to test whether this pathway is also present in other types of viral cancers. The program organized various activities, including symposium, seminars, working progress, and inter-lab meetings. The Cancer Center adds value to the program, which including state-of-the-art shared resources. Many of the vignettes that I have presented to you have used these as shared resources. Other value including pilot projects, support for leadership, and faculty recruitment. The program also adds value to the Cancer Center by promoting scholastic interaction among the cancer biologists and provide unique expertise, such as the use of virus as a tool for therapy and experiments. The program promotes inter-program collaboration, showing here some published examples on KSHV, EBV, HPV, MCV, innate immunity, and immunotherapies. For the catchment area research, we work closely with community outreach and engagement and other programs to examine HPV prevalence in the Western Pennsylvania and head and neck cancer patients. We have also examined the prevalence and risk factor of infection of polyoma viruses. We have several ongoing clinical trials in clinical cancers. And finally, the next cohort is the longest HIV cohort in the United States and has recruited a quarter of the subjects from the Pittsburgh catchment area. Example study of using this unique population, including examination of soluble biomarker for inflammation and T-cell activations, and characterization of Treg and CD8 T-cell in HKS patients. As a new program leader, my future plan, which is closely aligned with the vision of the Cancer Center, is to promote team science by focusing on our signature research areas. Specifically, we will recruit HBV and HPV virologists. We have recently succeeded in recruiting Dr. Haitao Guo, who is an expert in hepatitis B virus and viral therapeutic, as a program co-leaders. Dr. Guo's expertise fill a void of the program. His main role is to provide leadership in viral therapeutic as well as promote training and monitoring. The two of us will work closely on all the major decision-making for the program. We will continue to recruit HPV virologists with translational emphasis. This new recruit will be able to work with the community outreach and engagement, other program, Women's Cancer Institute, and head and neck clinics. We will take advantage of our clinical cohort, particularly the MAX cohort, to identify new biomarker for AIDS-related malignancy and non-AIDS-defining cancer. We will also take advantage of our expertise and focus on the mechanism of persistent infection of large and small DNA viruses. In particular, we would like to focus on RNA biology, take advantage of our genome center. These would include viral-encoded long non-coding RNAs, circular RNAs, and RNA modification. We have developed some specific plan, including we hope the completion of this plan will lead us to a submission of a program project by the end of 2020. And finally, we would like to promote the training of next generation scientists. I would be happy to take any question that you might have. Thank you.
All right, Dr. DeMayo.
So I have a number of questions. You say in your aims that you're a highly collaborative group, but you have 8% intra-programmatic collaborations. What might explain that, or how are you planning to address that?
So intra-program collaboration rate is low. The reason might have to do with the specific expertise of each member, because every one of us tend to work on a specific virus. They already have their own expertise. So the collaboration tend to reach out to either institutional or inter-institutional that reflect also the high percentage of inter-institutional collaboration. And obviously, there's some sheer area that we have identified, such as I have shown in the future plan. Persistent infection is present for large and small DNA virus, which are the expertise that we have, as well as RNA biology. A number of groups are working on it. So we think in this area that will facilitate further collaboration, and that might build up some more intra-program collaboration, including the submission of PPG.
The critiques raised a number of questions, and you didn't really directly address them in your talk. So for example, they said about clinical trials. What clinical trials has your program participated in or helped get started?
So there's a number of aspects that we participate in this area. So the critics is the review recommends to increase interaction with the clinical investigator. So first of all, virus we think are excellent target for therapy, including immunotherapy. So number of clinical trials are based on this, that the identification of this target, and then we connect with the CII program as well as the CTP program that build on that, and then we have some new clinical trial. Second area is the oncolytic virus is also our major area of research. A number of investigators has also worked on this area and work with this clinical investigator, which allow us to translate to the clinical trial. And there's also epidemiology and outreach area that I have mentioned that we have shown published work that we work with a community outreach. And so those might not be clinical trials, but they are translational clinical studies.
I would suggest you might want to emphasize one or two of those, since those are specific critiques.
Okay.
I'll address them.
So I just briefly, I guess I have to a little bit more specific.
Yeah.
Okay.
Do you think it's likely there are new additional major human tumor viruses? That's a major goal, thrust of your program is new pathogen discovery, which you didn't discuss here, but you did quite a bit in your write-up.
So that's a subject for discussion that within the field. And my personal view is that it is possible existence of other cancer virus, although many investigators have extensively mined the TCGA data set, but there's some limitation in terms of the number of cases or type of cancer that's available tend to be the major cancer, so ignoring those minor type of cancer. Secondly, for association of specific cancer virus, you need a specific population to have a to identify as causal law. At best case is the MCV identification. You really need to define epidemiology and have unique population to study it. And then, of course, there are some specific mechanism that might mediate this process, such as hit and run, and you might not be able to identify the sequence in this tumor, or such other case similar to microbiome. So the virus community is also out there, and they might also promote cancer progression. The question is, do you have the right population to study it or not? So these will require the development as well as collaboration with epidemiologists, as well as technology advancement. I think that remains to be seen.
And one last question. The catchment examples you gave, some of them, to me, were not compelling. One of them was you're doing clinical trials in your catchment. Well, of course, all clinical trials are primarily in your catchment. That's where the patients come from. So I would not emphasize that. I think you also mentioned looking for new polyomaviruses, but at the end of the day, is you didn't see any, but I understand it, correlation in your population. So I would take that out and try to focus on things that I think are more clearly catchment related. quite a bit has to do with HIV and KSHV. You might get some pushback on that. Are the rates of HIV and KSHV infection high in your catchment? If they are, you should say that.
Right. So this is part of the study whether either prevalence or risk factor, there's some specificity, specific association in the catchment area here. Basically, that's the whole basis of this study. So,
All right.
Yeah, well, I had many of the same questions that Dan did, so those questions are likely to come up at the site visit. But an additional one is your list of program activities. Where in that list are the activities that actually help you design and maintain your strategic vision going forward and align it with the Institute? You have listed a lot of seminars and symposia, but where's the sort of activities around planning?
okay, so what I have listed in that activity that has been ongoing, those are more in the past 5 years. Going forward will be in the future plan that we would like more focus on team science, as well focus on the area of our expertise, and as well as take advantage of the existing resources, such as the clinical cohort, the genome center, and that actually fits into the team science expertise, such as RNA biology, persistent infection that across all the virus that we are studying. So, so those are the specific action plan that we are doing. But also, we also want to recruit some other cancer biologists can further strengthen this interaction. So we have recruitment, actual action, and as well as some pilot project from the cancer center that can support this activity.
So following up on Dan's questions, how do you determine cancer relevance of the grants that you included in the program and the funding? Because you gave them all 100% cancer relevance. And I notice you have many R01s on reovirus. You have a R01 on chikungunya virus. And I would ask what's your rationale for 100% cancer relevance for those?
Perhaps two areas a little bit more specific that you refer to, such as the reovirus as well as the HIV, perhaps, because the other, I guess, is more straightforward. They are direct cancer
Right,
relevance.
but I think you're going to be challenged to ask why you included a chikungunya virus 100%
Sure.
cancer relevance. So I think kind of
So-
focus on answering that, not the HIV ones.
So there's for reovirus as notavirus, there's two aspects. Number one is some of these viruses are some of the best vector for oncolytic virus. And actually, so the group is doing some very fundamental basic study that allow us to design better oncolytic viruses. That's number one. Number two, innate immunity, as I mentioned in one of the vignettes, is closely related to cancer progression and inflammation. I think the basic mechanism will allow us to better understand the cancer progression and inflammation. And for HIV, I actually think it's easier to justify because many of the investigators, they have those grants. They actually directly study some of the AIDS-related malignancy, such as Chuck Rinaldo. He himself has been studying KSSV and immune response in the last 20 years and have many papers using advantage of his max cohort. And I guess Dr. Merrill, which is a newly added member, he has long track record of AIDS HIV clinic, and himself studied AIDS-related lymphoma, which is one of the major cancer in AIDS patients. And in the same time, more importantly, this clinical cohort allow us, other investigators, to collaborate with them. And one of the examples is recently we are studying biomarker using the pilot project from head and neck sport grant to fund that. And that's actually, without this clinical investigator, we would not be able to do this type of study.
So I think that's a strong response, but realize if you have a reviewer who who only claimed 50% cancer relevance
Yes.
and was more conservative in the assignment of the grants, they may take you to task on that.
Do you actually have the opportunity to assign a percentage? Yeah, so I think in those cases, in those particular ones,
Yeah,
it's
they're,
not
50% or
most
25%.
would claim 30 or 50 on a chikungunya
Yes.
virus grant, not 100% cancer relevant. So just be ready because it'll open up looking at a lot of the portfolio. You don't want that.
Perhaps another thing you might mention is, in that topic, is that the inflammation and
Yeah.
those issues that these viruses cause may be relevant to immune response to tumor viruses.
Okay.
I mean, the
And
advent of immunotherapy and immunology and inflammation has made many of these, that even 10 years ago, you would have been lucky to get 10% relevance. So you
And I
can
actually
swing that a
can-
bit too, but I'm just, be ready for the question.
Well, I can actually add some specific example that we link inflammation to cancer progression, such as in the case KSH-induced cancer, we have shown complement activation, Toll-like-4 activation. Those are more bacterial-like or other type of typical innate immunity, but they are closely associated KS progression, at least in our model system. And then this has been published, and I add those to strengthen the argument too.
So I just have a general comment. I didn't read the write-up, so I'm just going based on the presentation that I heard. But my sense is pretty much what Dan and Ed said, is that the strong science in this program and the impact is not really coming through. I'm trying to figure out why it doesn't sound as strong as I think it is. The figures, I think, are still a little complicated. Try not to have more than one science, one figure as part of the slide. It also may be the impact statements. Sometimes, and this should be true of all the programs, impact statements should not simply be a restatement of the conclusions. Impact statements are how it changed the field, whether it led to a clinical trial or led to a biomarker or it led to a discovery that other people have built upon. I mean, the impact statement should not just be, we concluded that X, Y, or Z. And that might help, but the science in this program is really strong, and it's just not coming across as one of the top viral oncology programs in the country, which it is. So I'm still trying to figure out how to get it to that point so that the science examples, and it may be because I didn't read the write-up. The write-up may have gone through enough detail that it would come across that way. So those who read it, might.
Yeah, I think the obvious historic strength has been discovering 2 new tumor viruses. That's fantastic. I think that time is largely past, and I think you're a new leader, you want to get across the new exciting things that are happening. I mean, I'm not even sure I would show the slide of the history, for example.
Part of it, we try to have a compromise, not talk too much about the senior investigator, including Dr. Chang Mo and myself, more talk about the junior investigator. So many of the vineyard intentionally pick those junior investigators. So there's a...
No, I think that's fine, but you have a slide of the history of the program, which I don't think any other program has. I'm not sure that's a slide you need to show.
Thank you.